In this video, I would like to show you a reviewer that I made back when I was in 12th grade. Essentially, this was just last year. I made this, I remember, back in October of 2023 because a few days after the first quarter exams, eventually I took the USTET. So what happened was I spent more time preparing for the first quarter exams, which included this topic. This is basically all about biochemistry, more specifically about cellular respiration and relevant topics. Um, as a consequence, I wasn't able to prepare enough for the USTET. In the Philippines, that is the University of Santo Tomas entrance test. Usually, well, at least based on my observation, UST is the second best university in the Philippines when it comes to medicine, which I initially intended to pursue. Who knows, maybe I still might pursue it in the future. But for now, I'll stick with ChemEng at De La Salle University. Anyways, let's start. Um, yeah, just wanted to say briefly before we start officially. Um, I placed a lot of dedication in making this reviewer. Although my capacity isn't limited to this, I just wanted to show you my creation. One of my best creations so far. And yes, for a forward, this is not complete, definitely. Um, according to the Philippines version of the education curriculum, um, there's definitely going to be an insufficiency. I won't be able to cover all of the topics, definitely. And when it comes to specific definitions, uh, as long as it's not written here, I cannot guarantee that I'll be able to explain that. So whatever is here, that's all I'm gonna say, for the most part. Okay, so let's officially start. So for the modes of ATP synthesis, when you say synthesis, um, this is basically the creation of ATP. What is ATP? This is adenosine triphosphate. So it's the basic unit, I guess. I think you call it unit. Unit of energy for a cell. So there are two types. We have substrate level phosphorylation and oxidative phosphorylation. They're different, definitely. This one is used in, for the most part, it's at least in this lesson, it is evidently used in the process of glycolysis. So the 10 steps of glycolysis will be discussed later. 10 substrates and as well as the final product, which is intended to be used for the next step, what happens after glycolysis. By the way, what I'm talking about is a process. So this is called cellular respiration. There are 4 steps. Glycolysis is the first. So substrate level phosphorylation, that's used in glycolysis. For oxidative, this is mentioned in the 4th stage of cellular respiration. More specifically, the electron transport chain and chemiosmosis. More on that later. So, if two, let's say substrates, I think that's the right term, I'm not sure. If two somethings are considered isomers, they have the same molecular formula, but at the same time, they have different molecular arrangements. So, let's go here, just to show you what I mean by isomers. For example, here in step 2 of glycolysis, what's mentioned here is that glucose 6-phosphate and fructose 6-phosphate are isomers. What does that mean again? So if two, such as the two substrates that I mentioned, are considered isomers, that means they have the same molecular formula, but they have different molecular arrangements. What does this imply? Based on my recent research, uh, this is for better adaptability for the overall process. But I believe there's a better answer as to why this is relevant, or what's the significance of two substrates being isomers. Because definitely, not all substrates in each step, because each step involves two substrates. For example, for step 1, glucose and glucose 6-phosphate. Definitely they don't, not everyone are isomers to each other. Only step 2 so far. I haven't mentioned anything else. So, aside from step 2, going back, let's go here. So glycolysis is a 10-step process. It's divided into two parts, two phases. The energy investment phase, or EI phase, and the energy payoff phase, or the EP phase. So yeah, I can't really explain properly this illustration. Let's see what I can do. So from here, glucose, 2 ATP is used. Keyword for EI phase is that 2 ATPs are used. So the result is 4 ADP and 2 phosphates. Why is that? Well, ATP has 3 phosphate groups. If we use them in the EI phase, 2 phosphates are, like, in a way separated from them. So as a result, we have 4 ADP and 2 phosphates. Now, in the energy payoff phase, it is intended for 4 ATPs to be formed. So as a result, also 2 NAD plus, 4 electrons, and 4 H plus, they turn into 2 NADH and 2 H plus. I'm not sure if it's reduction or oxidation. That's the appropriate reaction as to why, for instance, 2 NAD plus becomes 2 NADH. More on that later. Actually, let's search it. Okay. The answer to that is here. This, by the way, is the Krebs cycle. More on that later. But first, let's go here. So the reduction is the gaining of electrons. Let's see. Oxidation is a loss of electrons. Okay, so I think we're talking about a reduction reaction here, because by the phrase gain or acceptance of electrons, you have an increase in negative, you remove a plus. And I think that's what happened here. This plus got removed. I think this is a reduction reaction. So the final product in glycolysis would be 2 pyruvate and 2 water molecules. More on the final products later. Okay, so for the net products of glycolysis, so we have glucose turns into 2 pyruvate and 2 water molecules, as stated earlier. We have the formation of 4 ATPs, but in the EI phase, or the energy investment phase, again, 2 ATP molecules are used. So what we have left would be 2 ATPs. Now, we have 2 NAD plus, 4 electrons, and 4 H plus. This turns into 2 NADH and 2 H plus. Okay, sorry about that. Okay, so let me just show you what it looks like for the entire illustration. Let's go here first. So glycolysis is the first stage of cellular respiration. Electrons are carried via NADH only in glycolysis. But for the next stages 2, 3, and 4, electrons, well, more specifically only 3 and 4, electrons are carried via NADH and FADH2. The reason why we have FADH2 is because something happened in the citric acid cycle. More specifically, FADH gets involved. Where do we find that? Here. So this is the citric acid cycle. So from succinate to fumarate, FAD becomes FADH2. I believe this is a reduction reaction again, although there's no plus sign, so not sure about that. But yeah, FADH becomes part of the picture. In the other highlighted parts, NAD plus becomes NADH. So yeah, I'll be explaining that more later. Okay, so let's go here. In glycolysis, one glucose becomes 2-pyruvate through the degradation process. I can't really explain this. Okay, never mind. I remember. So stages 2, 3, and 4 of cellular respiration are done inside a mitochondrion. This again is the singular version of the plural mitochondria. We all know that this is called the powerhouse of the cell. That's where ATP is generated. It kind of makes sense because majority of the ATP that's generated happens here, inside the mitochondria. Although it's not impossible for ATP generation outside the mitochondrion, more specifically in the cytosol. And that explains why glycolysis happens in the cytosol and there is a production of 4 ATP, but because we invested 2 ATP, we have 2 ATPs left. And yeah, anyways, next, substrate level phosphorylation. This is the process in which ATP is formed by the transfer of a phosphate group from substrate to ADP. So yeah, we'll be using the term substrate later many times when we discuss the glycolysis steps, 10 of them. Okay, so some steps in glycolysis and the citric acid cycle are redox reactions. So basically, dehydrogenase transfers from substrates to NADH plus 3 times. So FADH2 1 time, this is in step 6. What are these steps? What are we referring to in terms of the steps that are mentioned? Again, that's in the citric acid cycle. Actually, what I mentioned to you earlier, this is step 6. So as you've seen earlier, redox reaction happens here. NADH redox reactions happens 3 times. Steps 3, 4, and 8. I think it's the same numbers shown earlier. Steps 3, 4, 8. Yes, that is correct. Okay, so let's move on to pyruvate oxidation. So acetyl-CoA, well, you might ask, how does pyruvate, the 2-pyruvate coming from glycolysis, become acetyl-CoA? Well, there's definitely a reason for that. Let me just double check. Okay, so it's a pretty lengthy process. Not too lengthy, but there's quite a lot of details. One of those details is that CoA connects with the pyruvates, so it becomes acetyl-CoA. That might not be as important as the other details that I'll be mentioning, but yeah, you might want to consider searching on that too. So the 2-acetyl-CoA, from here, there is a breakdown of glucose to carbon dioxide. So CO2 produced by cellular respiration represents the fragments of oxidized organic molecules. What else do I have to mention for oxidation pyruvate? Keyword: oxidation oxidized organic molecules. I'm not sure if that's a correct way of remembering it, but might help. Okay, so from pyruvate oxidation, we go to the citric acid cycle. ATP, so how is ATP generated again? It's through the specific kind of phosphorylation, that's the substrate level. So this is a process in which ATP is formed by the transfer of a phosphate group from substrate to ADP. So if you add a phosphate group to ADP, what used to be diphosphate will now become triphosphate because of an addition of one phosphate group. So it explains why it becomes ATP. Okay, so by the way, CO2 in citric acid cycle, here we have H2O. These are essentially byproducts of their corresponding stages. So for citric acid cycle, carbon dioxide is a byproduct. For electron transport chain, water is a byproduct. So another detail that you have to know is that for stages 3 and 4, the same stages that I've been referring to since earlier, they do not require oxygen to function. They are anaerobic, but it doesn't mean that even if there's oxygen, they won't function either. They would. Regardless if there's oxygen or not, they can still function. That's the bottom line. So, electron transport chain, this is, in other sources, the third stage of respiration. Maybe this is called third stage if we do not consider glycolysis as a first stage. Because technically, what makes this different is that the rest happen within a mitochondrion. Okay, so there is an acceptance of electrons from the breakdown of products from stages 1 to 2. So these electrons are passed from one molecule to another, and so at the end of the chain, the electron transport chain, electrons are combined with oxygen and hydrogen ions, resulting in the formation of water. Again, water is a byproduct of ETC, or at the fourth stage. So, the energy released at each chain step is stored sorry. Is this from or form? I'm dyslexic. This is funny. The energy released at each chain step is stored for the mitochondrion to use to make ATP from ADP. Okay. So this is where the other type of phosphorylation comes in. Earlier, we're talking about substrate level. Now, we're talking about oxidative. I believe both types, regardless, produce ATP, but only in different ways. So, oxidative phosphorylation is the production of ATP in the ETC. Substrate level, it happened evidently in the citric acid cycle as well as in glycolysis. So, oxidative phosphorylation is the process in which ATP is formed as a result of redox reactions in the ETC. Okay. I think this is already discussed. Let's go to the next page. Okay, going back to stage 1, glycolysis, the 10-step process. So again, it's divided into 2 phases, the energy investment phase and the energy payoff phase. So let's talk about the energy investment phase first. So this is where we consume energy. This consists of steps 1 to 5. For step 1, from glucose we go to glucose 6-phosphate. So all of the boxes are essentially substrates, just to let you know. And everyone that are encircled are enzymes. They're intended to, well, more specifically in the cases of steps 1, 3, 6, 7, and 10, well, except 6. As you can see, with the red ink, there is an indication of phosphorylation. So again, let's go back to the meaning or the definition of phosphorylation. So this is the process in which ATP is formed by the transfer of a phosphate group from the substrate. So what substrate are we talking about? So for instance, for step 1, glucose is the substrate. So through the enzyme hexokinase, the phosphate group from glucose gets transferred to the ADP. Okay, maybe that's a wrong example. Here, in step 7, so this is a substrate, 1,3-bisphosphoglycerate. So coming from here, what do you call this? The phosphate group from this substrate gets transferred to the ATP. So yeah, I'm not sure if that's the correct explanation, but something similar to that has to be valid. Okay. It doesn't go directly to the ATP. So from the substrate, it goes to the ADP. And with the addition of the phosphate group, it becomes ATP. I think that's the correct explanation. So, why did I say that step 1 may have not been the correct example? Well, look, in steps 7 and 10, it says substrate level phosphorylation. So it only applies in these two steps. Why not step 6? Well, why did I highlight it just like the rest? Well, this one is different. It's special. This is the only step wherein we see the production of NADH coming from 2 NAD+. So a redox reaction happens here. A reduction reaction. So, how about steps 1 and 3? Why doesn't it say substrate level phosphorylation? Perhaps it's because this is the energy investment phase after all. So coming from ATP, you turn it into ADP. As a consequence, the substrate from glucose, I'm not sure how to explain this properly, but so from glucose to glucose 6-phosphate, maybe in the let's say the substrate in the final product of step 1 is given a phosphate group. So that's the reason why it says 6-phosphate. So there's an addition of a phosphate group. So from there, let's go with step 3. So here, fructose 6-phosphate, we add another phosphate group to it, coming from the ATP, which makes it become ADP. Our final substrate becomes fructose 1,6 biphosphate. So instead of just 6-phosphate, we have 1 and then we have the prefix bi. So I think that's what's happening. And yeah, what does it mean again? If two substrates are isomers, such as the one in the case of step 2 as well as here in step 4. I just saw this now. What's the relevance? Well, what's the meaning? So again, if two are considered isomers, they have the same molecular formula and different molecular arrangements. Okay. I won't be mentioning the specifics anymore. It may take time. You just have to read it. Okay, step 5, isomerase catalyzes a rearrangement reaction. Is this a reason possibly as to why for step 5 there is a feature of being rarely reversible? I'm not sure about that. But definitely there's a reason as to why this one is rarely reversible when it's already the last energy investment phase. So this is basically the final product of the AI phase. So this is what happens at the end of the AI phase. That is the production of 2 glyceraldehyde 3-phosphates. So that one is going to be used in the energy payoff phase. So in step 6, this one, first things first, 2-NAD plus becomes NADH. We're given, I suppose, a phosphate group. I think that's the case. So sugar is oxidized, turning or reducing NAD plus into NADH. Energy is released from the said redox reaction. This is exergonic. And this is used to attach the phosphate group to the oxidized substrate. So, okay. So we basically add another phosphate group to the substrate that is already oxidized. So this makes the product increase potential energy. So step 7, substrate level phosphorylation. This happens in steps 7 and 10. We produce 2 ATP each, and that's the reason why we have 4 ATPs produced, as said earlier. In step 6, we have the formation of 2 NADH, but because we spend 2 ATPs in the EI phase, the end product would be 2 ATPs only, because 4 minus 2 is 2. And yes, we have 2 NADH. What is this? Dehydration reaction. Not sure what, I can't really explain this part. Kinase. Kinase enzyme is involved in adding the phosphate group reaction. So I think this detail can help you memorize the enzyme for steps 7 and 10, because as long as there is the prefix, I guess you call that prefix, I don't know. As long as there's a word kinase, then that should already imply that that step is either step 7 or 10, and it involves the production of ATP by adding a P group reaction. And that can explain why the ADP becomes ATP. Okay? Believe glycolysis is finished. What's the next step? Next page. Okay, so I think this is the net reaction for glycolysis. So glucose plus 2 NAD plus 2 ADP plus 2, what's PI? I'm not sure what this means. Let me just double check. I don't know what this means. Inorganic phosphate. Okay. Anyways, I won't delve to this detail anymore. This is inorganic phosphate, I think. This one's 2 pyruvate, 2 NADH, 2 ATPs, 2 hydrogen or H plus, and 2 water molecules. So phosphorylation is the addition of a phosphate group to a substrate. What is this? Step 4, DAP, G3P. I'm not sure what this means. 2 G3P molecules enter the payout phase. Oh, wait. Is G3P an abbreviation to this glyceraldehyde 3-phosphate? Kind of makes sense because it says G3 and then P. Okay, I think that's one way to memorize. Maybe you may not be required to memorize the entire term, but at least the abbreviations, such as this and this. Okay, what does this say? 2 ATP molecules were consumed in steps 1 and 3 of the EI phase. 4 ATP molecules were produced in steps 7 and 10. This is because everything is double in the payout phase. Makes sense, because what does it mean if everything is double in the payout phase? 2 2, 2, 2, 2, 2, 2, 2, 2. So yeah, where did the trend start? Here. So in step 5, we started to have 2 G3Ps. So further, 2 NADH produced in step 6 of the payoff phase. So if you're asked what step were the 2 NADH produced, that's in step 6, or the first step of the payoff phase. Okay, oxidation versus reduction. Oxidation is the removal of hydrogen and the addition of oxygen. Reduction is the addition of hydrogen and the removal of oxygen. Simple as that. Okay, citric acid cycle. Okay. I might make a separate video about the 4th stage. That's definitely difficult to understand, to recall, because again, this reviewer is made more than a year ago. Could have been easier if right after the exams I made a video out of it already, but that wasn't the case. That did not happen. I was busy for those steps. Anyways, Krebs cycle, where do we start? Oxaloacetate. I think that's the first step. Okay. So, starting here, oxaloacetate, so from oxaloacetate to citrate, what happens is glucose and fatty acids, again, they create acetyl-CoA from what was mentioned earlier. And the CoA is removed. What the result is citrate. So citrate synthase. This might be an enzyme, I'm not sure. So water comes in and it comes out. We have isocitrate for the 3rd step, aconitase. So step 3, this is where NAD plus, again, steps 3, 4, we have the creation of the NADHs. Isocitrate is oxidized, reducing NAD plus to NADH. 1 glucose is equal to 2 acetyl-CoA. That's therefore equal to 2 cycles. I believe from there, it is implied that the Krebs cycle happens twice. Not just once, but twice. And the net product, we have 2 ATPs, 6 NADH, and 2 FADH2. Why is it 6 NADH? Because, well, that's because in steps 3, 4, and 8, NADH is produced. So each step produces 2. So that makes 6. FADH2, we have 2 of them in step 6. Okay? Alpha ketoglutarate, well, before that, CO2 is released. Again, in the Krebs cycle, or the citric acid cycle, CO2 is a byproduct. It's released. CoA enters again, CO2, another CO2 releases. This one is isocitrate dehydrogenase. This one is alpha ketoglutarate dehydrogenase. I'm not sure if these are related to dehydration reaction, if that might be relevant. No, I'm not sure. But here it says enzyme dehydrogenase removes the hydrogen atom from the substrate. Again, what does removing a hydrogen atom mean? Essentially, removing a hydrogen means oxidation. Oxidation, isocitrate is oxidized so that NAD plus would turn into NADH. So oxidation, that's the key word. Okay, succinyl CoA, GDP becomes GTP. CoA is released again. I'm not sure what this means, but is there an explanation somewhere? Okay, GDP is guanosine diphosphate. GTP is guanosine triphosphate. What's the difference though between ADP and GDP as well as GTP and ATP? I'm not sure. But in stages 1 and 3, there's a transfer of phosphoryl group from the substrate to GDP or ADP to form GTP or ATP. This is the same definition we had earlier, but only what makes it different is, well, we involve GDP and GTP, but the concept is still the same. So for the succinyl-CoA synthetase, I'm not sure where that belongs. Maybe that belongs to step 5. But in step 6, we have this. So FAD becomes FAD2, succinic dehydrogenase. So it stays in the inner membrane, not in the mitochondrial matrix for some reason. Succinates hydrogen are transferred to FAD, forming FADH2. I think oxidation is also the case there as to why FAD gets converted into FADH2. So fumarate through the enzyme fumarase becomes malate. An H2O molecule enters. Okay, so H2O plus fumarate is equal to malate. That's basically the math to it. And from malate, when it's oxidized, because of this, NAD plus becomes NADH as a consequence. In other words, malate dehydrogenase. There is a reduction from NAD plus to NADH, and this results in the regeneration of oxaloacetate. Okay, going back here, alpha ketoglutarate loses a carbon dioxide, as you can see here. Succinyl-CoA is oxidized. Or do you see that? Is it this? So when succinyl-CoA is oxidized, there is a reduction of from NAD plus to NADH. I think that's the case. So it's easier to memorize what happens in between alpha ketoglutarate and succinyl-CoA. So yeah, I'm just not sure though if this is the reason or effect, whether these two are what these two are in terms of being cause and effect. But yeah, they're related, that's for sure. How about here at the center? So to oxidize acetyl-CoA to 2 molecules of carbon dioxide, oxidation again is the loss of electrons, which will be picked up by electron carriers NADH and FADH2 for the electron transport chain. So essentially, the citric acid cycle isn't the step wherein you produce the most ATP, but it gives way to the production of the electron carriers, or specifically 6 NADH and 2 FADH2, so that the electron transport chain would generate a lot of ATP eventually. So yeah, it's a complex process, but it's very fulfilling to understand. Okay, going back here. Okay, so reduction is the gain or acceptance of electrons. Okay, so FADH2 and NADH are reduced variants. If they're oxidized, they're NAD plus or FAD plus. More specifically, this one is nicotinamide adenine dinucleotide. FAD is flavin dinucleotide. CoA is coenzyme A. Oxidation is the loss or the pass of electrons and hydrogen ions. Okay, again, gain of electrons and hydrogen ions. Loss of electrons and hydrogen ions. In step 6, FAD gains 2 electrons and 2 protons in order to be FADH2. For steps 3, 4, and 8, NAD plus gains to be NADH. I think I missed writing parts here. Maybe there's a reason why. Not sure. But yeah, that's all I'll be explaining for this part. What's next? Okay. This one is intended to be skipped. I didn't think that the exam would be very specific in terms of each step. But what I did mention were definitely part of the exam, especially knowing the difference between oxidation and reduction and how they are related with NADH and FADH and their reduced counterparts. No, oxidized counterparts. Even I'm confused. Usually get confused. It is confusing at first, especially for someone who just studied it again. Anyways, GTP functions to make ATP molecules or directly power work in a cell. So definitely, any work that's done in a cell requires ATP. So results of the citric acid cycle. Per cycle, what's produced is 3 molecules of NADH, 1 molecule of FADH2, 1 ATP, and 2 molecules of CO2. This is a byproduct, and then this is given off. In what specific instances do we find the production of 2 molecules of CO2? Definitely not just step 3. Let's go back. I think there's a mistake. I suspect there's a mistake. So CO2 are released. I suppose steps 3 and 4. But yeah, I forgot to write 4 there, I think. But I won't change anything in the reviewer. It's, I guess, vintage at this point. I don't know. But yes, the right answer is still correct. Just a correction here. It's steps 3 and 4. So, this is only per cycle. You might wonder why earlier I mentioned values that are twice these numbers. Again, because, what's the reason again? Because we have 2 acetyl-CoA, there are 2 cycles. So 2 cycles, that means we double what's produced in 1 cycle. So per 2 acetyl-CoA is equal to 2 pyruvate is equal to 1 glucose molecule. So NADH, we have 6 of them. FADH, we have 2. ATP, we have 2. CO2, we have 2. Okay. Okay. Step 5 represents the only ATP generated in the citric acid cycle. Let's see. Yeah, step 5. So this is where we see the production of the only ATP. 1 ATP per cycle, but we have 2 ATPs because, again, 2 cycles. That's the standard. In nature, it really was intended to have 2 cycles, not just 1. Okay, more on this in the next video. I'll still have to study how this works. It's not an easy topic, definitely. But yes, essentially the ETC is where you produce a lot of ATPs. Um, this part of the process I still kind of remember. This is the smallest turbine known, living turbine in the universe, or at least what we know so far. And yeah, this is where a lot of ATP is generated. I'll be explaining this as well in the next video, and yeah, and the rest. Thanks for watching. See you in the next video.